Nice. Okay,
There,
let
there.
me just make you a moderator. Okay.
Hello. Can you hear me?
Yeah, how are
Yeah,
you?
I can hear you.
Good. How you guys?
Great, great. Yeah. Happy to have you here.
Yeah, I think it's gonna be fun.
Definitely, definitely. So, okay. We got Colin Ewald in the house. Great. Okay. It's 4 o'clock, so let's just begin because we are on time. So today we have the great privilege of having Kristen Fortney joining us on the Longevity Biotech Show. Kristen is the CEO and co-founder of BioAge Labs, one of the most interesting longevity companies out there. They use AI and machine learning techniques to find anti-aging drugs. So we'll get more into that later. Just some housekeeping things. We're going to start off with maybe 30 minutes of prepared questions, and then afterwards we'll open up for audience Q&A. And if anybody in the audience comes up to ask a question, just FYI, we are recording this and posting it later on YouTube. So just yeah, keep that in mind that we can use if you come up. That means you consent to your voice and I guess photo being showing up in YouTube videos. So let's just start right away with some introductions. So I'll go first, and then Robert, you can go second, and then Kristen. So myself, I'm Nathan. I'm the editor and author of the Longevity Market Cap newsletter. It's a once a week sort of roundup of different developments in the longevity industry. And I'm also the founder of Longevity List, which is a website where people can go find jobs, companies, and investors in longevity biotech. So,
So yeah, thanks. I'm Robert. I work currently at the Aging Biology Research Lab based short distance outside of Montreal in Quebec, Canada. And I have about 10 years of experience as a bioinformatics and research software engineer in both academia and industry. In the last year or so, I've been getting more involved in aging biology research proper and in the longevity biotechnology scene.
Hey guys, Kristen Fortney, co-founder and CEO of BioAge, a clinical stage biotech company developing drugs that treat aging and diseases of aging. And before starting the company, my background is in aging biology and machine learning at Stanford and the University of Toronto.
okay, great. So, one of the reasons why we wanted to have Kristen on the show was for one reason was basically we're trying to provide sort of a roadmap based on her experience building BioAge, so that other founders who are interested in getting into longevity biotech or the anti-aging uh, drug discovery space, that they might have a sort of template or a way to learn from her experience. So maybe we can just start off with the present. So Kristen, what is BioAge and what are you guys doing right now?
Yeah, sure. So right now, well, I guess what is BioAge? And really we're built on some pretty special human data sets because humans age on the scale of decades. I think it's important to have molecular data that span decades to understand aging. So we've made a bet on investing heavily into building some really unique aging cohorts. So we have blood samples collected up to 50 years ago when people were healthy and middle-aged that have electronic health records tied to those samples, tracking those people for their entire lives. And that means that you can go into those samples and you can measure everything with modern technologies, the proteome, the metabolome, the transcriptome, and see what predicts the future, right? See what predicts future longevity, see what differentiates those people who go on to live 90 plus in great health from the rest of us who don't. What's different at the molecular level. So we really see as BioAge is being built on top of this molecular map of human aging that we've built. And what we're focused on at BioAge is sort of going after those targets, one after the other, and focusing first on those targets emerging from our map that are the closest to translation. So we're actually focused first on in-licensing drugs that already exist that hit targets in our aging map, where we can go immediately into a clinical trial in older people. And just over the past year, we've brought in our first 3 such assets, clinical stage assets, and we just kicked off our first phase 2 trials very recently.
Yeah, yeah, the news was amazing. I should congratulate you. I forgot to, but yes, congratulations on bringing 2 phase 2 trials to the clinic, which is incredible because I think most people don't even realize that this is happening, right? These are drugs that BioAge has identified as potentially, modulating aging, and they're being tested in humans in phase 2. And I feel like more people should know about this. But yeah, it's really cool that you guys are doing this, and congrats once again on getting to this stage. It's incredible work that you guys are doing. But yeah, going back to BioAge. So you guys are leveraging these biobanks data samples, right? I think this is really interesting that they have this. Can you tell us a little bit more about what's in this biobank?
Yeah, for sure. So it's really longitudinal samples collected over time, and it is blood samples because that's pretty much all you can get from a healthy person. And these were collected when people were still healthy and middle-aged, and then over and over again for the rest of their lives. And tied to each of these samples are really detailed health records that have information on how long people live after their samples were taken, what diseases they ultimately got when they were older. But also, some of our cohorts have great health span variables too. So at every subsequent exam that these people went to, they measured things like their walking speed, their grip strength, their cognitive function. And we're very interested in looking at the molecular differences that predict not only lifespan increases, but also health span increases.
Yeah, I think that's really important, the measurement of cognitive function, because health span is really important. You can live very long, but if you have cognitive impairment, the quality of life, if you have, these sort of diseases, neurodegenerative diseases, can be very low towards the end of your life. So I think that's really important. Maybe we can go back now because this is a little interesting. We've covered what BioAge is doing now, what you guys, your approach, but let's go back to the beginning to trace your path to the present. So maybe I can start by asking you, what were you doing right before you started?
Right before I started, I was a postdoc at Stanford working on the genetics of exceptional human longevity. So, studying what's different about the genomes of people who make it to 100 or 110. And there's a common theme here, right? Like, I think the low hanging fruit in the longevity space is to copy what's already working, because there already are some humans who are living really long and really healthy, right? So, we know that it's possible. And there's a bunch of great examples. One of my favorites, you might have heard, Leela Denmark. She was a super centenarian, so lived to be over the age of 110. But she was actually a practicing pediatrician until the age of 104. So that means, fully mentally capable, physically capable. And I think it's really exciting that, it's possible to achieve that within, normal human variation.
Yeah, I think that's a good point because a lot of people in the longevity community are like thinking really far ahead, and they're saying, oh, let's extend maximum lifespan, which would be a good thing too. But even just as you said, the low hanging fruit, if we can just increase people's median lifespans, because average lifespan is maybe 80 years now, and just looking at the maximum, there's a huge gulf between the median and the maximum, and all the different things that we can do to improve health span. I think that really is the low hanging fruit. But okay, so you were a postdoc at Stanford, and then what sort of inspired you, or what was the catalyst to say, okay, hey, I'm going to start a company?
I always thought it would be a really valuable experiment to see what you could measure it midlife that would predict ultimate longevity. And you can get biomarkers from that, you can get drug targets from that. And I would say there were sort of two different kinds of scientific findings that inspired the idea. One was, this was sort of, this was, I guess, only a couple years after the great methylome work by, Steve Horvath and Trey Eidecker had come out, showing that from these genome-wide data sets, you could build great predictors of chronological age. So just, I'm not sure if everyone's familiar with that work, but prior to these discoveries, people had looked at a handful of, quote unquote, biomarkers of aging, like telomere length, that had very low predictive power for your age or your future health. And the fact that if you looked at these sort of genome-wide data sets and got all that predictive power out of that, that was, point one that was really exciting to me. The other finding that I thought was really exciting was some early work of Richard Miller, where he'd done these lifelong experiments in mice, and he'd measured just a handful of things at midlife, things like their T cell counts, et cetera, and showed that they predicted mortality. And I wanted to sort of combine those two ideas, right? So let's build predictors not of your chronological age, which you can already measure, but let's build predictors of mortality. And I thought that would be a way both to find biomarkers and identify interesting drug targets. So that's kind of the scientific inspiration.
Okay, great. Yeah. So you had the inspiration for BioAge, but then how did you actually go about uh, building something that would be investable? Because I remember in one of your interviews, you were talking about how a lot of the early work in this kind of approach for a biotech company could be done on, just your laptop. And I think this is really a powerful idea because a lot of people who are potential co-founders in longevity biotech, they're thinking, oh, I need to, set up a lab or like, have a lot of money and put it into my startup before I can even get started. But maybe you could dig a little deeper into this idea of virtual biotech and CROs and this kind of stuff.
Yeah, for sure. I mean, especially for bioinformatics right now, right? Like, I mean, there is so much public data out there that you can build to identify and test hypotheses. There are, exomes for tens of thousands of people through the UK Biobank. There's all this public genetic, and there's microarray data for millions of samples on all these human diseases. So there's an awful lot you can do with just your laptop. And I think the CRO landscape, which you also mentioned, has also been evolving very rapidly to the point where there are a lot more virtual biotechs right now. So you don't have to actually make a huge infrastructure investment, buy a lot of your own equipment from the get-go. You can just pay to have some proof of concept experiments done to de-risk further work.
Right. So when you started BioAge, and you were trying to get to your, I guess, first proof of concept or something that you could pitch to investors, um, uh, can you share like how much money you had to put in, like your own money into the company? It's okay if you don't want to share particular, numbers or whatever, but I'm just curious if you.
Oh, sure. Yeah, it wasn't that much. It was between five and $10,000, I think, that I'd put in personally, and this was sort of why, well, I was a postdoc, and and mostly supported by data analysis.
That is amazing. Because I think the general idea when I put this question out on Twitter, I got many responses saying, okay, you need at least $100,000 to get started. But yeah, this is like an order of magnitude smaller, which is a much more achievable goal for people who are, I guess, straight out of grad school or postdocs or stuff like that. So that's really encouraging. Um, so okay, so then you have started BioAge. You had this idea. Then the next step, I guess, was you found this data set or this biobank data. Where did you go from there? Like, how did you go about de-risking and then figuring out how to, I guess, expand and possibly take investor money? Maybe you can tell us about these inter.
Yeah, for sure. And just to mention too, like I think there is a lot of seed and pre-seed money available for promising science that could have potentially a huge payoff. Geography does matter a lot, right? So BioAge was funded in Silicon Valley, and um, I came from Toronto. That's where I did my PhD, and probably wouldn't have been able to raise there because it's just, very still location dependent at the time, and hopefully that's changing. But it's still true that there's tremendous benefits to being, located in certain areas and working on cool science. Um, so BioAge, yeah, it started off as really just sort of this hypothesis that if we built these data sets, there would be all this signal there for really future longevity. And this is kind of bold, right? Like it's saying that I can look at, your proteome, your metabolome, and I will see signals of something that is happening 20 years later, 30 years later, and the sort of phase one of the company was going out and partnering with the right biobanks, and even just finding the right biobanks because it's pretty rare actually. Like everyone is biobanking today, but there are not a lot of people who are biobanking healthy samples decades ago, as well as following up those individuals for the rest of their lives. So sort of finding those samples, getting them profiled, and showing that there was signal there. That's sort of like phase one.
Okay, great. And then I guess phase two, when you started to get traction and pitching to investors, I'm really curious about this because BioAge, if I remember correctly, was started maybe at the end of 2015. So I guess that was, 6 years ago or whatever, 5 years ago. And back then, I'm guessing longevity wasn't as big as it is today. What was the experience like pitching this idea of like treating aging with drugs and this aging biomarker stuff? Like, how did investors take this? Did they think you were crazy or?
Yeah, great question. Oh, and just for clarity too, so this phase one of actually profiling all these samples from the biobanks, that was done with seed money. So this was after raising seed for BioAge. And like I was, you're right, like it was a lot earlier in the longevity space. It was still after Calico. So Calico's big announcement with all their funding had been I think pretty recent in history. And I think that was probably critical to getting BioAge funded because people were becoming aware that there was this huge opportunity here. And there was at that time, there still is this sort of growing sense that the science is ready and it's time for translation. And there's all sorts of different investors in biotech and in longevity. And I raised from tech investors for the seed, and I think they appreciated the fact that we were not going after a particular mechanism, a particular target, but really taking a data-based approach that has the potential to find, multiple different mechanisms.
Right. And so it seems like um, tech investors, especially in Silicon Valley, the Bay Area, are very interested in longevity. I feel like traditional pharma, like definitely like big pharma, uh, they're still slowly coming onto this. But you did announce recently a partnership with Amgen, which is really cool. Maybe you can tell us about this strategy of repurposing drugs that are already out there as like your low-hanging fruit. And then maybe you could also follow on with your strategy with the partnership with HitGen doing actual novel um, compound.
Yeah, for sure. So just to add to your comment, yeah, I think pharma and traditional biotech, they don't know what to think about aging yet, right? I think once there's like a first clinical success for a drug that is definitely targeting a mechanism of aging, that's going to change everything. But we're not there yet. And in the meantime, I feel like there is a lot of low-hanging fruit just because there are a lot of things that are going to impact aging, and frankly, very few of them have been tried in the clinic. So our philosophy is always low hanging fruit, easy things first, one impossible thing at a time. So we did make, we built our map of targets on top of our biobanks, and we are deliberately going after, those mechanisms that are the most de-risked. And that means there's already a drug out there that has been through phase one that we know is safe, that hits its target, And we want to bring in all of those that we think might work and go, as quickly as we can to a pretty lean, pretty efficient clinical trial, because the clinic is a risky place to be. So I would like us to have a lot of different mechanistic bets where we can get a human efficacy signal before too long. And to your second question, we did recently announce a partnership with HitGen. Um, if I had to guess, like how many things we were going to in-license over time that might work, I would say, maybe around 10, certainly no more than 20. And then after that, I think it's important to move on to novel targets. So this is an important point too, right? So where the field of aging is today, I feel like we know an awful lot about how animals age. There are hundreds of things you can do to a worm to make it live longer, or a fly, or a yeast, most of which probably don't matter for us because the species are so different. But again, like a first way to use our platform, our human data platform, is as an overlay on, everything, all aging biology that is known, right? Because if there's, a gene you can knock out that makes a worm live longer, that by itself doesn't excite me that much. But if that same gene has an ortholog in humans with a very strong association to mortality, well, now I'm really excited, right? So I think that those are the easy targets to go after, those ones that have both a human and an animal signal for aging. But then I do think there are going to be a lot of important mechanisms for aging that are human specific. Like even mice, black 6 mice, which are the, inbred mice that most biologists use, they die pretty much exclusively of cancer. They don't get Alzheimer's on their own. Heart disease is not a bottleneck to their survival. So I think that the things that are the most important for their aging are going to be different from the things that are most important for ours. And while we're focused at BioAge, most of our focus, 80% I would say, is on in-licensing, we do want to start to explore some of the human-specific targets emerging from the data as well.
Cool. Yeah, that's great. So maybe coming back to these clinical trials. So you have two phase 2 assets. One of them is targeting unexplained anemia, and the other one is looking at, I guess, immunosenescence in COVID-19 patients or elderly COVID-19 patients. So it looks like the name of the game for longevity biotech companies is to choose these sort of low-hanging fruit indications first, because obviously, the FDA does not have an improved indication for aging. But what do you see as like the regulatory pathway going forward to bringing some of these drugs to, uh, I guess I would say the general population, people who are aging, which is everyone, but don't have like a defined sort of disease or age-related disease. So yeah, I was just wondering what you think about that. Like how do these sort of drugs get to people like?
Yeah, for sure. So a few points there. One is that while the FDA doesn't recognize aging as a disease, I don't really see that as a bottleneck to progress. Because if they do, when they do, I should say, it'll probably still involve a huge trial that where people take your drug for a long time, which is just, amounts to a lot of money, right? And for a mechanism that you're taking to the clinic for the very first time, you don't really want to bet the farm on that mechanism. So I think it always makes sense. It will always make sense to go into an initial proof of concept indication where you can be lean, where you can, have a trial with not too many people. That's not going to take too much time. You'll get that first human de-risking signal quickly. And that would be true even, I think, if we were a diabetes company, because those trials can be pretty long and expensive as well. And the second question about how does it ultimately get to everybody, right? And there's a few examples you can point to, and I think that ultimately you'd want every aging drug to follow a development path, something like a statin has, right? So those kind of are prescribed like aging drugs right now. They're given to people who don't necessarily have disease. They're over 40, they have a couple of biomarkers, risk factors, and they're prescribed statins. So I think that that's a historical precedent, and there are some examples like that. And how statins, of course, were first approved is that they were first tested for a very narrow indication, familial hypercholesterolemia, and the indication was widened over time. And that's a similar kind of path, I think, that we will have to take with our mechanisms.
Okay, yeah, great. Okay, so we're rounding up to almost half an hour. So I just want to ask a couple of quick questions before we open up for audience Q&A. So do you have any sort of words of advice or maybe things that potential longevity biotech founders could do or not do? Maybe that's more important when they're thinking about building these a company in longevity.
Well, first of all, definitely go and found a company in longevity. The space needs more founders. There's so much low-hanging fruit. I would love to see more people getting in there, and there's so many opportunities that just need good people driving them. In terms of things to do or not do, those are always long lists. I mean, just, de-risk at every stage, I guess, right, is the good advice, and make sure that you survey the landscape and know. I would say too, don't necessarily, like a lot of people I think who are coming in not from biology, trust the literature too much. So step one is if you read something exciting in a science journal, you definitely want to replicate it in your own hands before you commit to it.
Yeah, that's a really good point. I've seen some pretty brutal paper retractions when I did my research on various aspects of longevity. Okay, so maybe just another quick question before we open up for audience Q&A. Is there anything that we can do to help you right now? I know you previously, posted some jobs, so maybe you're hiring. I guess maybe you're not raising money right now because you just recently raised your Series C. But yeah, if there's anything that we can do to help your mission, or anybody in the audience can do to help your mission, I just give you the floor right now to.
Yeah, for sure. Yeah, so we're not fundraising, but we are doing a ton of hiring. And it's always, it's great when everybody is aligned on the mission. So we like that a lot at BioAge. So definitely go check out our careers page. There's lots of different roles that we're looking for right now for people with science backgrounds, computational backgrounds, clinical backgrounds.
Okay, great. So everybody who's listening to this right now in the audience or afterwards on YouTube, go check out the careers page on BioAge if you're interested in joining an awesome longevity biotech company. So yeah, I think we're basically there. Oh, maybe I'll ask one other question since I had a little bit of time. So Reason, the co-founder of Repair Biotechnologies and also the author of fightaging.org, he will periodically write a request for longevity startups. So just like a blog post about things that are maybe overlooked in the longevity industry that he hopes that other startups would pursue. I would wonder what you think about this. Like, do you have any ideas or requests for startups in the longevity space? Maybe not specifically therapeutic, but maybe some sort of infrastructure thing that you think would really help either BioAge or just the longevity biotech industry.
That's an interesting question. I've got a whole long list of things. Well, I mean, point one is that like pretty much everything exciting in longevity science is currently not being translated by anybody. So I think there's a lot of opportunities out there, with the possible exception of senescence, which I think there's a lot of activity around that specifically. Not that we've had traction yet, right? So even there, but some of the, I would say, particularly underfunded, potentially high impact areas are I think organ transplantation, xenotransplantation is a really exciting area that can do things at a larger scale. I think you mentioned infrastructure as well, and I think a lot of people outside the field don't know how old it is to purchase old mice. Because ultimately, if you have a longevity therapy, you've got to put it into an old mouse, an old animal to see if it's working. And you literally cannot buy these because no one else is a customer, you know? And so why would, you know, Charles River or Jax or these mouse providers even age them that long? So that's, you know, something we built out ourselves at BioAge. We've got a bunch of really old mice, but I think that's a barrier to a lot of other people in the field being able to go in and test their hypotheses efficiently. So just, you know, on the that's an important component that I would love to see someone develop a CRO there.
Yes, I totally agree. I've thought about this a lot as well because I remember looking at some of these prices at the Jackson Lab, or I think the subsidy at the NIA, and it's like $300,000 to do sort of like a mouse lifespan study, and I think that's extremely
It's
extremely
nuts.
prohibitive. But yeah, it's crazy. It's totally crazy, and I think someone will eventually crack this problem. I think like Mousera slash IAM was like an initial sort of foray into this problem. But I think someone else will figure it out eventually with maybe automation or something like that. Or at least I hope so. Okay, so it's 4:30 now. Just to reset the room, we have Kristen Fortney here with us, CEO and co-founder of BioAge Labs, a company that leverages AI and machine learning to find drugs that modulate aging. So without any further ado, I guess I'll switch it over to Robert. I think he had a couple of quick questions as well, and then we'll open up for
Yeah, thanks, Nathan. So, Kristen, nice to have you here. Just a couple of questions. These could easily take, you could have whole separate calls for these too, but um, maybe you can just share your quick thoughts on these. So, I got one hard question and two uh, possibly also hard questions. Which one do you want first?
You said they're all the same, so whichever you like.
All right, I don't know which one's which here, so I'll start with the easier of the hard questions, I guess. So in another interview, you were on Jake's the Pod of Jake interview or podcast some time ago, and you mentioned Greg Egan as an inspiration for why you got interested in longevity and that kind of stuff. Can you just share for like a minute, like what was it? Which one of his books? How does the current longevity scene square with where you see this going, like in the science fictional direction? And yeah, just a bit more about that.
Yeah, for sure. So yeah, I've always been interested in aging, and I've always been a, you know, an avid sci-fi fan. And Greg Egan in particular, or early Greg Egan, I should say, was very good at making reasonable assumptions and then sort of following them through. I like a lot of his work. Maybe Diaspora is my favorite. But there's a lot of, yeah, I mean, sci-fi in general, right? There's a lot of boundaries, I think, that we can, you know, transcend with science, and that's exciting, and we should do that.
So Diaspora is the book that that you like the most of his.
Probably my number one favorite. Yeah.
Okay. The next question I had was about AI in drug discovery. So uh, Bruce Booth, for example, from Atlas Venture, partner Atlas Venture and blogs at LifeSci VC, he has an interesting post from a couple years ago called "4 Decades of Hacking Biotech and Biology Still Eats Everything," something along these lines, where he essentially argues that despite all of the advances in computer-based approaches and big data, high throughput, it's still just one piece of the puzzle. Do you think that maybe the AI angle is perhaps a little bit too hyped up sometimes? And like how would you answer the critics or the skeptics in this area?
Yeah, for sure. I think it can be too hyped up, but honestly, I think that that is just how you do modern science now, right? Like if you really want to know what's going on in a human or another animal, you have to be looking at genome scale things, whether it's the genome itself or the proteome, the methylome, the metabolome. And these are data sets of a scale where you can't just stare at them and figure out what's the most important. Of course, you use statistics, and of course, you use machine learning. And I think that these tools are going from being, overhyped, a decade ago, maybe even 5 years ago, to being kind of in the water, because that's just how you analyze these data sets at all. Um, I think there has been a bit of a backlash in the past because some people dove into the AI side of it without carrying that through to the biology. Like biology is still really hard, and there's a lot of constraints imposed by what you can actually test, what you can actually do, how much you can understand actually about a biological system. But I guess I feel like the field is past that now, and that these are just, the tools of modern biology, or will be, the established tools of modern biology in a couple more years.
All right, fair enough. And my last question has to do with research roadmaps. So the folks behind the Healthy Longevity Challenge, if you could actually go to healthylongevitychallenge.org, which is affiliated with the U.S. National Academy of Medicine and some other major organizations around the world, they're working on a global roadmap for healthy longevity. And also there's something at the NIH called the Bridge to AI, part of the Common Fund. And also Eric Morgan, your co-founder, is listed as co-author on a paper discussing identifying biomarkers for biological age. So I guess I'm just wondering if you're aware of or involved, if BioAge is involved beyond that in any way with any of these roadmaps, like trying to work with the rest of the longevity research and biotechnology community and biomedical research more broadly.
I'm not aware of those particular initiatives, but we are involved in some other industry initiatives to try to build. I mean, I think this is really important for BioAge and for other companies as well, right? Like for example, our first drug, which we're testing in the anemia of aging. That first indication is not aging itself, but we are, giving drug to an older population with functional deficits for 3 months. So there might be something going on there. And I would love to have a biomarker that told me if we were rejuvenating people, beyond just, uh, looking at the primary outcome for the trial. And so we are building for that. Like, for example, in that trial, we're doing full proteomes before and after treatment. And that means that we can, compare it to our human cohort data. Everyone has, a clinical trial, pared down version of a Fitbit as well. So we'll be sort of collecting continuous movement data. But just to, so that, just to be clear, the cohorts that we've built, I think, are also really useful for building biomarkers of aging, right? Because you can actually build a predictor of incident mortality, something that predicts, how many years of life are left, right? And that's exactly the kind of marker that you can then use to assess whether the people in the trial are being rejuvenated to some extent by the drug. So we, I don't think we have things that are ready yet, like it's a beta version, but I'm very excited by that area of aging biology, and I think it's important.
Awesome. Looking forward to what BioAge will come out with in the near term. All right, Nathan, back to you. Those were all my questions.
Okay, great. Thank you, Kristen and Robert. Okay, so we're going to open up for audience Q&A. If you raise your hand, we can let you up to the stage. Just FYI, we are recording this, so if you come up, that means you consent to us using your voice recording and I guess photo recording of your photo on the app. So while we're waiting for people to raise their hands, maybe I'll just ask another follow-on question to Robert's last question. So this whole idea of having a biomarker that sort of predicts, I guess, mortality, how do you see the roadmap for something like that being accepted by the FDA? Because I feel like, if you look at the history with statins and using, I guess, cholesterol as a surrogate biomarker, that could take some time. I'm just wondering, because I don't really know the answer, but what you think about how long it might take for something to become accepted.
Yeah, that's a great question. I think these things take way too long. I mean, hopefully we'll see bone mineral density being accepted as a biomarker for osteoporosis as well. But importantly too, right? These things like cholesterol and bone mineral density are, I think, actually have a lot less predictive power than some omics biomarkers that could be built. But that's just such a long process to get these things approved. So I guess I feel like they're going to have scientific value and de-risking value for a company in the very near term. Um, but in terms of when they'll actually be approved as a surrogate endpoint, yeah, I think that's a long way away.
Yeah, and it's just like I'm trying to visualize how it's going to happen because it's like, hey, we have this biomarker, or it would be like some sort of, composite biomarker, and you're going to tell the FDA, hey, we have this neural net that predicts mortality. This is the thing that you're going to use for the clinical or primary outcome measurements or something. It's just like I feel like it's going to take some convincing.
That's right. Yeah, the near-term stuff that I still think could be really important is I think less ambitious, but still could be really important for the field. Like for example, today, if you wanted to run a clinical trial for frailty, the approvable endpoint you would have to use is a 6-minute walk distance. And that scares everybody off from doing development there because a 6-minute walk distance is incredibly noisy. It's incredibly dependent on who is administering the test. It's just this one-off thing that you measure, right? So I am more optimistic that in the near term that, these continuous wearables signals where you're actually collecting movement data, over several months will lead to something much more robust that could become like a frailty pivotal trial endpoint. And that I think would be really important for drug discovery and development in that area.
Yeah, definitely. I definitely agree with that. Okay, awesome. Thank you for that answer. Let's take some audience questions. Daniel Ives, we have Daniel here. He's the CEO and co-founder of Shift Bioscience, another longevity biotech company. So
Hi, Kristen. Really interesting to listen to you. I just had two very different questions. So one is you're going to test sort of different low-hanging fruit sequentially. The question is, have you thought about testing it all at once, like basically like fruit salad? That would be the term. Just give it everything, like all the different targets. And the second question is, like personally, are you collecting blood samples longitudinally so you can map yourself to so the work you're doing right now?
Yeah, great questions. So I think combination therapies are ultimately where things are going to go. Um, but they are much more challenging from a clinical development perspective. And I guess I also feel that some of the individual targets we're pursuing will have, big, strong effects on their own, like we've seen for lots of different age-related diseases. So phase one will be sort of finding out the different pathways you want to modulate that will work to, by themselves. And then phase two can be, building combinatorial therapies of the things that work. And the second question is a great question. I haven't actually ran my own proteome, but I'd love to. Yeah.
Thanks, Kristen.
okay, Serge, what's
Hey
your
guys,
question?
thanks for inviting me to the stage. My question is what's the ETA for reversing gray hair? Thank you.
Yeah, interesting. I'm actually not an expert on that area, so I don't think any of the mechanisms we're pursuing would do that. You probably want some sort of stem cell thing oriented to scalp, but yeah, I don't have a good answer to your question.
Okay, John, uh,
Hi, Kristen. I'm a MD PhD with a PhD in artificial intelligence for bio, and I'm interested in some of the question that Robert kind of touched on earlier and some of the machine learning. And specifically, I was wondering what you thought of the potential of deep learning for drug discovery and maybe even graph neural networks. Just kind of what your thoughts are for these as having potential or too much hype for drug discovery.
Yeah, for sure. There's a lot of different parts of drug discovery where AI ML can come in and play a part. So for example, at BioAge, we're focused on using these tools really for target ID, and other people might use them to build molecules, and other people might use them to, inform clinical trials. We actually hope to do the latter as well in the future. And really, it's a matter of the data, right? So for example, like transcriptomic data, there are millions of profiles in the public domain. And what that means is that, yes, there's definitely a role for deep neural nets there because these are very noisy data in sort of the original representation, and you can, clean that right up with the right kind of framework, right? But for a lot of biological data sets that are still meaningful and important, there's just like maybe tens or hundreds of samples. I mean, there are some sort of transfer learning methods that might be useful there as well. But often for those data sets, simple methods will extract most of the value already, and it'll be sort of like a small return for building something a lot more elaborate.
Thanks.
Okay, thanks for your question, John. Guillermo?
Great, thanks, and thanks, Kristen, for your time today. You all mentioned briefly how difficult it is to think through appropriate surrogate endpoints and just the challenges that are sometimes very specific to aging in terms of clinical trial design. But I was wondering how you all are thinking simply from a preclinical standpoint about de-risking and validation. And it's probably so disease specific that I know maybe it's hard to answer, but I was just, I guess, curious whether you're looking to innovate not just on the discovery side, but also, or how you're thinking through the sort of de-risking and validation side from, in vitro and in vivo assays.
Yeah, that's a really good question, actually. So we've been very deliberate about our choices there as well, because we're doing really our initial pathway discovery in humans, but then you want to verify causality, right? So you want to go into an old mouse and change something and see that it's doing what you hope it's doing. And so many mouse models of disease are not translatable at all. And so we made the deliberate decision to focus on indication areas that were very translatable from human to mouse, that happen in old mice just like they happen in old humans, and that were, a few other criteria too, right? There had to be good data on them in our human cohorts, et cetera. But for example, right, like that's why we like muscle aging, because old mice, just like old humans, they don't walk as well, they don't run as fast on their wheels when they get older. And that's basically like a human frailty trial. So that's an experiment, we love to do, where you take a bunch of old mice and you give them drug, and you can just have the wheels in their cage report automatically like how many rotations get done each day, and then you can see if over time, the mice on drug are doing better than the mice not on drug. And that, we think should be translational to the human scenario, both in terms of the biology, but also the measurement, right? And similarly with immune aging, because just like us, when mice get older, their immune systems don't respond as well to challenges. They're much more likely to die, in response to influenza or COVID. So that's one reason why we're not going after, Alzheimer's preclinical models.
Got it. Okay. Yeah, that makes a lot of sense. Then that's very smart. Thanks for that input or perspective.
Okay, thank you, Guillermo. Next question from Martin.
Hi, Kristen. This is kind of tied into the previous question. It sounds like from these biomarkers, you're going to be jumping straight into mouse models. I was just wondering whether you thought there was any value in cell models. So just looking at whether any of these biomarkers you identify individually can do things like extend replication times of cells or stop cellular senescence. Thanks.
Yeah, I think that's interesting. Actually, it's funny you mentioned senescence, because we're actually looking at blood, basically omics. So including blood proteomics. So we actually do see a lot of SASP type factors in our data, which I think is really interesting scientifically. Yeah, so far we've focused on having the in vivo validation as well. And if we're doing stuff in cells, it's more around the particulars of the target to see that, the downstream pathway is getting activated in the way that we think it should. Just because we want to be sure that it's going to have that organism level sort of degree of effect that we need for the trial. So we haven't, we don't have a lot of high throughput cell-based assays that we rely on.
Thanks.
Okay, Do we have any other questions from the audience? If not, maybe I'll ask another question. So, oh, sorry, we have another person here, but I'll ask my question first. So, Kristen, about about finding a co-founder. So maybe you can tell us a little bit about the backstory behind how you met your co-founder, how you kind of decided the division of labor. I know your co-founder Eric Morgan is also like a technical background, so there's this idea, I guess, that some people have in for biotech startups that you need one technical co-founder and one like business development co-founder. But I guess there's obvious examples where that's not really needed either. So maybe you could just tell us your thinking on co-founder selection.
Yeah, for sure. So Eric, well, I've known him for ages, right? Which is, it's always nice to have that pre-existing relationship with the co-founder. So I first met my co-founder Eric on the math team in high school, so a long time ago. And we previously worked together on even on aging papers when we were both at the University of Toronto. So it was really sort of very, very natural for us to go into this project together. And we do have some, I guess, redundancy of skill set, in a good way. I think we both care about the human data and, the statistics, et cetera, right? That's his background as well. He has a bit more of a medical background, I guess, and I have a bit more of a scientific background. But I don't know. I feel like you can sort of learn it on the go. I don't know that you necessarily need a very sort of well-defined profile type to get started. You just sort of want generalists, right? Who are going to dive into whatever needs to be, whatever the problem of the week is, right? And be able to execute on that. Yeah.
Yeah, I totally agree. Okay, cool. We have Dr. Anwar here. What's your question?
Hi, this is Anwar. Hi, thanks for letting me speak here in this group. I have a question about, I don't know if you've done this type of work, but have you looked into like time to event analysis in any part of your work, like survival-based analyses using machine learning AI models? And if so, then how did you go about evaluating predictors developed through machine learning processes such as like random survival forest type models?
Yeah, a lot of the models that we build are time to event where the event is death, but it can also be the onset of a particular age-related disease. And, we can evaluate them and compare them in the usual ways internally within a cohort, but we can also, build a model on one cohort and test it on another one that's from a different, ethnicity, geography, et cetera.
Right. I think so. I was asking more along the lines of, so when you looked at these predictors, how did you go about evaluating them for risk, using hazard ratios or any other metrics of those sorts?
You mean how do we build them? Is that the question?
No, how you went about evaluating the machine learning derived like important predictors.
Yeah, so that's by building a predictor in one data set and testing it in another, right? That's going to sort of
Right.
tell you how it does on independent data. Yeah.
Right. Did you evaluate the predictors like individually or together in terms of like any Cox model type analysis or um, any other types of metrics?
Yeah, the usual methods, which is Cox analysis, which is various flavors of sparse regression built on individual proteins as features or sort of more combined pathways as features. Yeah,
Right. Okay.
there's
Thanks.
lots of exciting models in the area that can be pretty much used off the shelf.
Right. Right. Thanks.
Okay, thank you for your question, Dr. Anwar. Michael, what's your
Hey, Nathan, another great session. Hey, Kristen, super interesting. So my question, a little bit higher level. So, in trying to understand the aging problem, there's like, the path of, like the hallmarks of aging paper and that whole line of questioning of just, what biological activities are going on that's causing aging to occur. And then, the other, like another line of inquiry is around like clocks and, finding the perfect biomarker to predict, biological age or mortality, as you kind of focus on, and, all of like the Horvath line of inquiry. Is there like another or two other kind of like buckets that you think about in terms of framing the aging problem, or are those two kind of like lines, holistic?
Yeah, I guess I feel like the hallmarks of aging paper is still very much a draft version, like I think it was modeled after the hallmarks of cancer paper, and that was sort of when we really understood what cancer was. And I think that for aging biology, it's still a guess. So I, and this might be a more unusual stand, but I kind of feel, I expect that there are a lot of mechanisms that are important for aging that are really not represented there. And part of this, I think, is because aging biology, most of the work in aging biology has been in invertebrates. It's been in yeasts and worms and flies because they live for just a couple weeks. So it's really easy to change a gene or give them a drug and see what happens to their lifespan. And not that much, has been done in mice, and even less in humans. So I guess I feel like those are important mechanisms, but there are going to be more. Um, and in a way, I think it's kind of unified to sort of, these genome-wide analyses, like you mentioned with the clocks, right? Like those are sort of asking in a data-first way, what are the changes that predict aging? And I think that those types of analyses might help us come up with, new mechanisms of aging to add to the list.
That's cool. So like you're saying clock research is kind of in service of finding new mechanisms.
Yes.
That's cool. Thanks.
Okay. Uh, if there aren't any other questions, maybe I'll ask another quick one. So there are some people who do work in sort of like uh, young blood rejuvenation or neutral blood rejuvenation approaches for anti-aging. And some people in this camp believe that aging is actually like a programmed sort of process, whereas I think the majority or consensus view, as I see it, is that aging is more of a wear and tear process. But I was wondering what you think about this personally, like your thoughts from like a high level. I know this is all kind of speculation, but what do you think about aging? Is it mainly wear and tear? Is there some sort of programmed element to it, especially, looking at, clocks and, blood?
Yeah, sure. So I don't think that it's programmed in the sense that the programmed aging people talk about it. Like at a high level, like of course it is, right? Because humans have a certain lifespan, which is different from trees, which is different from worms. So there is a sense in which, DNA constrains lifespan. But not in the sense that we usually talk about a programmed biological process like development, right? Where it unfolds according to the exact same schedule every time. Very sort of low variability in how it happens. And aging, of course, is incredibly stochastic. So yeah, I guess and there are species where it is programmed, right? And it looks completely different, octopus or salmon. Um, I do think that the young blood stuff is really promising work, but I guess I don't see it as connected to programmed aging.
Okay, great. Great. Okay, so we're basically at, oh, John, did you have a question?
I do have a follow-up, or not really a follow-up, but just a different question. Earlier, Kristen, in the talk, you mentioned lots of low-hanging fruit in the field. And as someone who's relatively new to longevity, I'm curious about this. And, Nathan has also told me similar things, that there's a lot of low-hanging fruit and kind of founders are the bottleneck. And I'm wondering if this is mostly in regards to like druggable targets. Maybe there are a lot of druggable targets out there that just need someone to pursue, clinical trials for a specific druggable target, or developing a new drug. But what just are some more examples of this low-hanging fruit?
Yeah, for sure. So there are, as you mentioned, druggable targets. There's a bunch of things that work in mice, that are worth trying in humans. And just sort of going down that list systematically, like most of those have not been tried. We haven't sort of tried to develop those to bring them forward to the clinic. There was this idea that Nathan just mentioned too around blood. There's a lot of evidence that rejuvenating the blood compartment is going to be an easier scientific challenge that should have, systemic benefits, right? Your blood goes everywhere. If you can make your blood young again, that can have a lot of good effects. And maybe you can do that with, a young bone marrow transplant, et cetera, right? So there's, I would say, the traditional sort of pharma type targets, that's probably the lowest hanging fruit because that's, very standard drug development. And there's a lot that hasn't been translated, but there's also these other interesting areas, around rejuvenating blood stem cells, around organ transplantation. Those are some examples.
Great, thanks. I agree. The rejuvenation therapy stuff is also very interesting. So thank you.
Okay. So we're at the 1 hour mark now. Maybe we'll close it out with just 3 quick lightning round questions. So these are questions for Kristen. If you can answer them in like a sentence or less, and then we'll just close out. So the first question is, how long do you want to live?
I don't not, I don't want to die, right? So there's my answer.
Okay, so as long as possible, maybe. Me too. Okay, number 2 is, what do you think about the Fermi paradox? Why haven't we?
that's a that's a different one. Yeah, I'm not sure, right? Yeah, I'm sure you guys know all the usual interpretations. Yeah.
Okay.
Hopefully, it
Yeah.
doesn't mean we're about to, kill ourselves soon, in some
Oh
massive
yes, yes.
extinction event. Yep.
Yeah, I hope it's not the great filter. That would be the worst answer.
Exactly.
Okay. And I guess last question. This one's a little new, but if someone gave you $1 billion right now and said, "Hey, go out there and accelerate progress in longevity," how would you allocate this kind of?
Oh yeah, great question. I mean, I have like a project list that I think is interesting. And if I had that amount of capital, I think I would go out and explore that space even more aggressively. I think there's a lot of people doing really interesting work in regenerative medicine and aging biology that are really just, limited by funds right now, where that could be a huge accelerant.
Great. Awesome. Okay, so once again, I'd like to thank Kristen for coming on the show. It was a great conversation. I had lots of great questions from the audience as well. And just a reminder, BioAge is currently hiring for many different positions. So if you're looking to join an incredible company in the anti-aging longevity space, they're backed by A16Z and a bunch of other really great venture capital firms. Yeah, I would definitely recommend you go check that out on their website. And without any, is there anything else that we want to cover? Oh yes, next week we have Matt Scholes of Oishin Biotechnologies, a senolytics company using gene therapy. He'll be on the show at our usual time, and yeah, I think that's it. I just want to thank Kristen again for coming on the show. It was a great privilege talking to you.
Thanks, you guys. Great conversation.
All right, take care.